Good afternoon, students. How are you all? I think everyone is fine. Okay, let us start the class, guys. In the today's class, we are going to talk about the principle of inheritance and the variation. Okay, this chapter is very important for the NEET exam, and in this chapter, we are going to get at least 4 to 5 questions. Kindly see here. The topics are very simple. Okay, it is not difficult. Also, very simple. 4 to 5 questions you are going to get from this topic, from this chapter. Okay, fine. I think you all know that what do you mean by inheritance, right, guys? Can anyone tell me what do you mean by inheritance? Before going to do that, can you tell me whether I'm audible and screen is visible, guys? 1 minute. Yeah. Sorry. 1 minute. Yeah. Good afternoon. Good afternoon, Nili, Helen, Navya. Can you tell me, guys, what do you mean by inheritance means? I think I'm audible and visible, right? Okay. Okay, just listen to the class. If you have any questions in between, you can ask me. Okay. You all know that what do you mean by inheritance, right? Inheritance is nothing but a transmission of the characters, right? From the parents to the offspring. Yes or no? From the, yes, pass of the genetic information. Very good. Pass of the genetic information from parents to the offspring is called as a inheritance, right? Some of the characters we also inherited from our parents, right? That is nothing but the inheritance. Okay. Then coming to the variation, you all know that variation is nothing but the change. Change, right? Variation is nothing but the change. We are going to get the change by the mitosis or by the meiosis. In the mitosis, the offerings are genetically identical to the parents, right? Whereas in the meiosis, offerings are genetically dissimilar to the parents, not identical. Yes or no? So there the variation came, right? Yes or no? So that is only we are going to study what is the principle that is behind exchange or the transmission of the character from the parents to the offspring, and what is the principle behind the variation which causing the variation in the offspring. Okay, fine. Let us start the class, guys. Okay. So first the experiment, the first question come to in the mind for the Mendel. Okay, Gregor Mendel. In the 1875, Gregor Mendel started the experiment and he took the sample that is the garden pea. He selected is the garden pea. Okay. I think from this slide, you are going to get the very small concept here is, okay. What you have to remember here is Gregor Mendel has taken the garden pea for his experiment and he selected 7 pairs of the true breeding characters. Okay, 7 characters he is chosen. Okay, which are those characters means stem height. How to remember means stem he is taking height and dwarf we are going to study, right? Stem height and flower color and flower position. Flower and pod and the seed. Okay, that you have to remember. Stem, that is a single. Okay, then flower, in the flower, flower color and flower position, that we know that axial and the terminal, right? And we studied violet color and white color flower, right? Then pod shape and the pod color. Pod shape and the pod color we are going to he studied, and the seed shape and the seed color. Okay, so this is the characters learned by the studied by the Gregor Mendel. So these are very easy to remember, but in the statement wise question they will ask you to confuse. Okay, so that's why you have to remember which are the pairs he selected for the exam for the experiment. Then coming to the Mendel experiment, guys. First Mendel experiment is a monohybrid cross. He took the plants that is having the true characters. One is a height and one is a dwarf. Okay, height plant, another one is a dwarf plant. And he observed that some factor is there. When the Mendel was there on that time, we didn't identify the cell structure and we didn't identify the these all the DNA structures and all the things, right? For that, he given some factors kind of name. Okay, factor. You know, all the Mendel experiment we can see the factor But now we know, right? The factor is nothing but gene, right? So here is taken the capital T and capital T and small t and small t. It is nothing but allele, right? Allele. Now you can see that capital T and capital T for height and homozygous tall. Homozygous means you all know that the same. It is having the same means capital T, capital T means we will call as that is a homozygous. Small t means we are going to call as homozygous. Okay. If it is having the capital T, small t, we are going to call it as heterozygous. Heterozygous. Yes or no? We are going to call it as heterozygous. The true breeding characters means it must have the homozygous character only. Okay, true breeding means. Okay. Then what will happen? The gamete will formation will take place. You all know that during the meiosis, see here, it is a diploid means. Okay, it is a diploid means after the meiosis, it will become haploid or diploid. It will become haploid, right? During the gamete formation is nothing but formation of the egg or the formation of the sperm, right? Once it is formed, it is having the haploid, right? It is having the haploid. You all know that when the gamete was formed, sperm is formed, right? When the sperm is formed, the sperm is carrying either X or Y, right? Like that, it is carrying T because it is a homozygous, right? In the female, what will happen? Both are same, X and X is right. So it will carry the X, right? So here what will happen? During the gamete formation, we are going to take a single one here. And because it is similar, no, that's why. And from T and small t, we are going to take this small t. And the F1 generation is having the tall. The tall means what? It is having the tall morphology, but the gene which is present inside it is the heterozygous. This condition we will call as a heterozygous tall. Heterozygous tall. Okay. Then what we are doing? In all the Mendel experiment, we are crossing this F1 with the F1 only. Okay, F1 with the F1 progeny only we are going to cross. Then we are going to get the ratio that is 3:1. Means in the first F1 generation we got the tall, but when we crossed it again with the F1 generation, we got that 3:1 means 3 tall and the 1 dwarf. And when we talk about the genotypic ratio, capital T is 1, whereas the capital T, small t is 2, and the whereas the small t and small t is the 1. So this is nothing but the monohybrid cross. Mono means what? We are taking the one character for the one character. One character for the hybridization. Okay. So this is a monohybrid cross. Just remember the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Okay. So you all know that in the F1 generation, we got that capital T and small t, right? But it is tall, right? But we don't know whether it is homozygous or heterozygous. It is looking like tall, right? It is looking like tall. But I want to know that whether it is homozygous, homozygous or heterozygous. Heterozygous means capital T or capital T small t. Now you tell me which experiment I can conduct for this to know whether the F1 generation is homozygous or heterozygous. Tell me guys in the chat box. To know whether it is a homozygous or heterozygous, which can I do? Whether the back cross or the test cross. Very good, Neeli. Very good, Simran. Very good. It is a test cross, right? For checking the homozygosity and the heterozygosity, I am going to do the test cross. Here in the test cross, what I am doing is, I am taking this heterozygous, that is a F1 progeny, right? And I am crossing that with the dwarf parent, dwarf, only with the dwarf parent, small t and small t. Then I will come to, then the ratio will come, that is the capital T and capital T and the small t. That is 1:1 ratio. Sorry, small t and small t, right? So that we are going to call as 1:1 ratio, one tall and the one dwarf we will get, right? So that is called as the test cross ratio. Test cross ratio for the monohybrid. cross is 1:1 for the monohybrid. What about the dihybrid cross, guys? Can you tell me what is the test cross for the dihybrid cross ratio? Dihybrid. What is the ratio of the test cross for the dihybrid ratio? If any students are from the YouTube, you can answer me in the chat box. The ratio of the test cross in dihybrid. In the MCQ, for confusing you, they will give you 1:1, 1:1:1:1, 1:2:1:1, like that. In that case, suddenly your mind will go to the 1:1 because test cross you studied is 1:1. Right. What about they're asking for dihybrid test cross? I'm not getting answer. This is a revision, guys. You already studied once, right? Can you think? Monohybrid means 1:1 means what about dihybrid? Dihybrid. Can I go? Can I only answer? Okay, remember, for the dihybrid cross, it is 1:1:1:1. Okay. Just remember 1:1:1:1 is for the dihybrid cross, test cross. Okay. Then we'll talk about the back cross. What do you mean by back cross means? We have got the F1 progeny, right? F1 progeny we have got. But this we are going to cross with any parent. Any parent. Any parent means either the dwarf one or the either the recessive parent or with the dominant parent. Okay, either with the recessive parent or with the dominant parent, we are going to cross on the time. Why we are going to do this? Okay, the ratio is not required for this. Why we are going to do this is to maintaining the parental character. In the usually in the agriculture and in the some of where we are growing the plant for the this purpose, right? For the hybrid character purpose, they will do the back cross. Okay, because the parental characters needs to be maintained. On the time they will do the back cross. Okay, remember this. In the back cross, what will happen? For the back cross, we will take the F1 progeny and we will cross with any parent. Here, why we are doing means for maintaining the parental character. Maintain parental character. Parental character. Okay, just remember these two things. Okay. And you know that test cross is used to know the unknown genotype. Right now, only I told. Okay. So now let us talk about the inheritance of the two genes. Now we will talk about the inheritance of two genes. We are talking about the two genes, right? So that's why we are going to call as dihybrid cross. Here mainly the Mendel has taken the two characters. Two characters we require. One is a round. Okay, one is a round character. Okay. Here for the round opposite will be wrinkle. Wrinkle. And here we are going to take the yellow color, and here green color. Green color. Raju, if it Raju is asking whether the back cross and the apomixis are same. Just see, Raju, back cross is a man-made. We are doing, right? We are doing. When you talk about the apomixis is happening in the plant only. You know, microspore mother cell is there deployed. That will not undergo the 1 minute. I will tell you here. Apomixis, you can see that microspore mother cell is there, right? It is without undergoing the fertilization or without undergoing the meiosis, it will divide. This diploid cell divide, right? It diploid cell divide by mitosis, mitosis, and by this it will give rise to the embryo. Embryo. So many mitosis means it will give rise to the embryo, right? But here in the back cross, what we are doing, it is a human made. We are doing that, right? Not plant itself is making, right? Both are different path. Raju, do you understood this? Do you have a question still? Raju, do you understood? Okay. Backcross and apomixis are not same. It is different. Apomixis is the it is like the asexual reproduction. Okay, asexual type of reproduction in the plants. Back cross, we are doing the hybridization. That is different. Okay. So let us talk about the inheritance of the two gene. So here we are taking the round and yellow character and the wrinkle and the green. Okay. This is the main progeny, main characters we are taking here. So round and yellow is given by round is capital R, capital R. Here wrinkle is R and R. And yellow is capital R, capital Y and capital Y. Here the green is small y and small y. Okay. So this is a pure breeding, right? Now we are doing the crossing. You know that from the capital R and small r, we are taking the one gamete. And from the small capital Y, we are taking the one gamete, right? And from the R, same gametes, no? So that's why we are taking the two different gametes. You have to separately round both the things because both are not same. Both are different gametes. Like how the during the sperm, the four sperms will be generated, right? During the meiosis, like that. So these are different. Okay. So here you can see that after the formation in the F1 generation, you are going to get the capital R, small r. So here how to you have to combine means first you take the round character, capital R and small r. Then here dominant is capital Y and small y. So now you have taken this. This is the dominant character it is showing, the round and yellow. Dominant characters are expressed here in the F1 generation. Then we are again crossing that with the, now again we are going to cross with the same F1 progeny. Then we are going to get this phenotypic ratio. That is phenotypic ratio. We can see that by our eyes, right? This is phenotypic ratio. That is 9:3:3:1. Okay. Can you tell me what is the genotypic ratio, guys? Genotypic ratio. What is the genotypic ratio? 1:2, 2:1, 2:4, 4:2, 1:2, 2:1. Okay. So this is the genotypic ratio of the dihybrid cross. Okay. So kindly make a note of this, both the thing. Both the genotypic ratio and the phenotypic ratio. This question will be asked in MCQ. Only the ratio. Okay. And in some type, you will get the question also to solve. Okay. And on the time, you have to put this Punnett square and then you have to study this. It will take a time. So that's why you have to do the practice of that. Okay. Fine. Then coming to the Mendel laws, guys, the Mendel has given the three laws. So here you have to know the difference between all the three laws. Okay. First one is the first law. First law is the law of dominance. Sometime you'll get the question from here also, which is the first law, which is the second, which is the third. Okay. The first law is the law of dominance. Here they will tell that the each gene is having the two allele, right? For example, capital T and the small t, right? These two allele, right? In this, the capital T will be dominant, one will be dominant, and another T will be a recessive. Okay. When these both are together, the dominant is going to express its character, right? That is going to be given by this law of dominance. The characters are controlled by the factors. Factors are nothing but genes, right? And the factors are occur in the pair. The factors are occur in the pair, that is the alleles, right? That is nothing but capital T, or small t, or capital T, small t. Okay, because they are occurring in the pair. Then in the dissimilar factors, I have told you, in the dissimilar factors, here the one will be dominant over the other factor. So here capital T is dominant overing on the small t. So that's why capital T is expressing its character. So that is tall. Okay. This is about the law of dominance. So here whatever they are telling, the characters are controlled by the factors and factors are occurring in the pair and dissimilar factor, one of the character is dominant on the other. This all are will comes under the law of dominance. Okay. Then in the law of segregation, they will tell only about the during the gamete formation. You know, during the gamete formation, these two alleles will be together, right? These two alleles will segregate separately, right? This is going to be tell by the law of segregation. For example, so here spermatogonia, you know that spermatogonia, right? Spermatogonia, the cell which is the cell which will produce the sperm, right? The spermatogonia cell is having the X and Y, right? But when it produce the sperm, it will produce the sperm, right? this sperm is having either X and this sperm is either having Y. Yes or no? This is nothing but gamete formation or during the segregation process. Okay. So here you can see during the gamete formation, either X or Y. Okay. During the gamete formation, the factors or allele. Here I have taken the example of X and Y allele. Okay. Factors or allele of the characters pair segregate each other such that the gamete will receive only one of the two factors. Yes or no? First, the X and Y both are there, but now after the gamete formation, it has got either only X or either only Y. Yes or no? Now, if it is combined with the egg, right? Egg, which is having the X means it is going to give a female. Yes or no? Do you understood this law of segregation? Nothing, guys. During the gamete formation, the two alleles are together, right? For example, I will tell you guys, you only now. Okay. Now, you both best friends are there, right? Now tell me anyone. Navya, you have any best friend in JNV? Best friend. While you are going for the mess, you will go together. Can you tell me name if you don't mind? Kalpana. Okay. For example, see, Navyashri and Kalpana are the best friends. Okay. They are always together. Okay. But what will happen? Navyashri is preparing for the NEET and Kalpana is preparing for the JEE. Okay. Now she got very good score in Kalpana got very good score in JEE and Navyashri got very good score in NEET now. Okay. After that, now both are segregated, right? Now Kalpana will go for if any NIT, IIT she got means she will go to there. And if Navyashri got any very good college means she will go to that college. They are segregated, but they are not now in Navodaya, right? They are segregated. Yes or no? But before they are together, now they are segregated. Yes or no? They are segregated independently, right? NEET student for the medical and the JEE student for the NIT and IIT, right? So that segregation is happening here. Now you understood, guys? Law of segregation, independently segregating both the alleles. You understood? Okay, fine, guys. Then coming to the law of independent assortment. Thank you, Nili. Law of independent assortment. Here, what will happen? Here, law of independent assortment. They are given this law by studying the dihybrid cross. By studying the dihybrid cross, the two characters, they have given this law of independent assortment. There we studied the ratio that is 9 is to 9 is to 3 is to 3 is to 1. Right? We have got that round and green also and green, sorry, wrinkled and yellow also. Yes or no? So here, the round character is not dependent on the color. Yes or no? Is that round character is dependent on the color character? No, right? We have got difference, right? Now we have got the ratio that is 9 is to 3 is to 3 is to 1 means we have got the difference, right? We have got how many type of things we have got? Round, yellow we got, right? And round, green also we got, right? And we have got the round, sorry, wrinkled, yellow also we got, then wrinkled, green also we got, right? Is that character is telling, is that color is telling yellow color, I will go with only round? No, right? The characters are independently assorting. Not, there is no two character that will assort jointly. Okay? It is assorting means it is segregating independently. The characters here. But whereas in the segregation, it is about the allele. Whereas in the law of independent assortment, it is about the character. Remember that. Okay? See here. When the two pairs of the trait are combined in the hybrid, two pair of the trait, that is round yellow and wrinkle green, if you take anything, okay? The segregation of one pair of the character is independent of the other pair of the character. Yes or no? So this is about the character. Law of independent assortment, it is about the character, whereas law about the segregation is about the allele and gamete formation. Remember this. I think you understood about the Mendel law of the inheritance. Let us go to the next thing, guys. incomplete dominance. This will not follow the Mendel's law. Just remember, incomplete dominance. Whichever the law we are going to study, right? These all are not going to follow the Mendel's law. Okay. First, we'll go to the incomplete dominance. First, we will talk about the incomplete dominance. Here, the two alleles will be there, right? Capital T and small t. Both are not completely dominant on the each other. Okay. For example, if we ask the small children, no, they are used to ask in the olden days, right? I don't know about now also. Who is your favorite? Whether the mother is your favorite or father your favorite? Yes or no? We'll ask these questions, right? If you find any babies, if you find any children like 1 or 2 years, right? The baby, if the baby used to tell that I like both, I love both my both mama and papa, right? Like that, here the gene, here what is happening is it is liking both the allele. Okay, it is liking both the allele, so that's why it is expressing both the characters. It is expressing both the characters. It is liking both mama and papa, right? Like that, it is expressing both the character here. It is an inheritance of in which the heterozygous offspring show intermediate character. Intermediate character. So what will happen here? Red flower is going to be crossed with the white flower. We are going to get the pink flower, pink color. Okay. So means here we are in the normal, in the Mendel thing, we are going to get the capital or smaller means we use it to get the red flower, red flower, right? But what about in the incomplete dominance means we are going to get the pink color. Okay. Then when we cross with the F1 progeny, F1 with the F1, we are going to get the ratio is 1 is to 2 is to 1. So here, both the phenotypic ratio and genotypic ratio both are same. Remember, genotypic ratio, phenotypic ratio is same in the incomplete dominance also and in the codominance also. In the codominance also, remember, codominance also 1 is to 2 is to 1 only, phenotypic and genotypic ratio. Okay, fine. Remember this, the question will be asked like this. If the red flower is crossed with the white flower and the progeny got the pink flower, which type of the inheritance is it? It is the incomplete dominance. Okay. Then coming to the codominance here, the inheritance in which both the alleles of the gene are expressed in a hybrid. Both are expressing. Okay. Both are going to be expressed here. Example we can take is the ABO blood group. In the ABO blood group, mainly you will get the how many genotypes you will get. How many genotype? How many genotype in the ABO blood group? Can anyone tell me how many genotype and how many phenotype? How many phenotype are there in the ABO blood group? Just remember the genotype will be 6 and the phenotype will be 4. That is A, B, AB and O, right? So these are 4 genotype. When we're talking about the, phenotype. When we're talking about genotype, for the A there will be 2, right? And for the B there will be 2, right? And remaining 1 only, AB and O. So that's why total it is 6. Because you know that A blood group, 1 minute. Yeah. You all know that A, oh, sorry, 1 minute. Yeah, here I will write. Here the A blood group, we can write like IA, IA allele also, whereas IA and IO allele also, right? So that's why A blood group is having the 2 alleles. Okay, remember this. And then coming to the, so here codominance means both are going to be dominant here. For example, if you take the AB blood group, here both A and B allele is going to be dominant here, right? So that's why the AB blood group is example for the codominance. AB blood group is example for codominance. Okay. Then coming to the multiple allelism here, more than two alleles of a gene governs the same characters. So here if you take the main character, here the multiple allelism means more than two alleles. More than two alleles are maintaining the one character. Okay, governing the one character. For example, a blood group is there, right? For example, blood group is a character. Blood group, right? Blood group is a character means for this character we have a three allele, right? IA, IB, and IO. right? I is there, right? So that's why the three alleles are there. More than two allele means we will call as multiple allelism. The ABO blood group is example for both multiple allelism and the codominance. Remember this. Okay, fine. So now let us go to the pleiotropy. Neeli Shruti, did you ask any question, pa? You have given yes, FHE 4. I didn't understood, pa. Do you have any specific question, Neeli? Okay, then coming to the pleiotropy, guys. Just remember you only, guys, okay, for the pleiotropy. So here, the single gene exhibits the multiple phenotypic expression. For example, if I am a biology teacher, now I am teaching only biology, I have to read only biology. But what about you? For NEET exam, you have to prepare for physics, chemistry, and biology. More, right? But you are single. But you are preparing for more than one subject, right? So here also same, the single gene that is exhibiting a multiple phenotypic expression. The single gene which is exhibiting multiple phenotypic expression, we are going to call as pleiotropic gene. Okay. For example, starch synthesis in the P and the phenylketonuria. If you take the phenylketonuria, if the phenylketonuria gene, that gene is going to be responsible for the color, pigmentation, right? Color and the mental retardness and the some other characters also. Okay, so that's why this phenylketonuria we call as pleiotropic gene, where single gene is controlling multiple phenotypic expression. Okay, and coming to the starch synthesis in the pea plant, you can see that capital B is the effective starch synthesis that will produce the large starch grain. Okay, for example, if you take capital B, okay, so this capital B is producing the large grains. Okay, and small b, okay, this small b, it is producing the small. Okay, but when we combine these two, it is going to give a capital B and capital B, right? And this is intermediate between these two. For example, this is intermediate between these two. Okay, it is intermediate between these two. Okay, so that's why it is also example for the incomplete dominance. Remember this. Starch synthesis in the plant is also example for the pleiotropic and is also the example for the incomplete dominance. Why means the result is the intermediate between these two, not the bigger one also, not the smaller one also. So that's why capital B and small b is the intermediate and also incomplete dominance. Remember this. Then coming to the chromosomal theory of the inheritance, guys. Chromosomal theory of the inheritance given by the first, you know that Mendel has published his work in the 1865, but it is unrecognizable because on that time we don't have WhatsApp, Gmail, email, and those all the things, right? So it is unrecognized. But the three scientists recognized that De Vries, Correns, and Tesma, they independently rediscovered that the chromosomals are vehicles of the hereditary. They told that, and the two identical chromosomes form the homologous pair. So remember here, whatever our Mendel has told, no? So same thing here, they're comparing with the chromosome. There we comparing with the factors, they are in the pair and they are segregating, right? But here the same thing they are applying for the chromosome. They're also together, there will be homologous chromosome will be there, and they're also segregating during the anaphase, right? The same thing they are telling. Okay, they're independently going to be othered. They're also in the Mendel's law of segregation, the gametes, the alleles are going to be separate independently, right? So that is compared with the gene allele. So here we are comparing with the chromosome, right? The genes are present on the chromosomes, right? Fine. So this is about that. So the chromosomal theory of inheritance, you can go through this also, but that much is enough. You have to remember the scientist name, okay? And the chromosomal theory of inheritance was scientifically, it is experimentally proved by the Morgan. Okay, it is scientifically or experimentally proved by Morgan. So here Morgan is selected the Drosophila for the experiment. Why is taken the Drosophila for the experiment means it can grow on the simple media and it can grow for within a 12 to 14 days only. And the progeny will be 100 to 1000 will be produced. So it will be in the 100. So it will be useful for studying. And the male and female flies are easy to identify. So that's why he selected Drosophila. This type also you will get the question in the statement wise. Okay. And coming to the linkage. So here we are told that while studying the Morgan experiment, he given the linkage. Linkage means, so if you take this the chromosome, on the chromosome, there is the two genes are located, right? The gene number 1 and this is the gene number 2, right? Here, the distance between these two genes, the physical association of these two genes is going to be called as linkage. Either they may be far or either they may be near. Okay. If they are near means we can call as linked gene. If they are far means it is called as unlinked gene. Okay. When we are talking about the linked gene, it will not give the no recombination will be there. No recombination. Okay. They will not segregate. They will not segregate independently. They will not segregate independently. Independently, remember. Okay. But what about the unlinked gene? The unlinked gene will segregate independently, okay? And this unlinked gene will give the recombination. Can you make a note of this, guys? This is very important regarding the unlinked gene and the linked gene. When they are together, they will go together only. They will segregate together only. There will be no recombination. For example, two friends are there. Okay. Now two friends are there here. What you will do? For example, yeah. Athene and friend will be there. Athene and Athene friend, close friend, are staying in the same apartment. Okay. Now they are going. They are linked. Means they are in the same apartment. Okay. When they are going for the shopping, they'll both go together only. Right. But now Athene's friend is there in the some other different town. Different town. Okay. They are like not linked, unlinked. They are far. Okay. Now what will happen? Athene want to go for the shopping means Athene will call to the person who is staying in the apartment or who is staying far away in the another town. Who is staying in the apartment, right? He will not call for the another person who is staying far away in the another town, right? So, like that, when they go for the shopping, they will choose a similar one only, right? When they go for the shopping, if they are going for the shopping, both of them will buy the same pant or same shirt, right? But when we are talking about that person is going for the shopping, right? He will choose some different because he is not with them. Yes or no? So that's why the recombination is nothing but a different. Okay. So just remember when they are linked, there will be a no recombination, no segregation. When they are unlinked, on the time segregation independently and recombination will be going to be happen. Okay. So recombination is nothing but generation of the non-parental gene combination. Okay. Okay, let us talk about the cross. Morgan has did the crossing in the Drosophila. So here mainly you have to remember, first he taken the body, yellow body. Okay, yellow body and the eye color. Okay, body color and eye color. Body color and eye color is crossing. Okay, for example, here this is a gene. So here it is the body color and here it is the, sorry, 1 minute. Now Morgan is crossing the gene. Okay. Now these genes are here. Okay. This is for the body color and this is for the eye color. Eye color. Okay. So what will happen? This is linked gene, guys. This is linked gene means they are together. Yes or no? They are together like the body color and the eye color. The body color he chosen is a brown body color, red eye male. Okay. And the female is choosing is yellow body and the white eye female. Okay. Now you tell me the genes are together, right? Genes are together now. They are linked. We are going to get the more recombination here. When the genes are linked or when the genes are together, are you going to get the more recombination here? I have explained in the last slide also, right? Yes, Neeli. Yeah, very good, ma. Very good question. Recombination will happen where it will not depend on the whether the gene is present on the same chromosome or whether the gene is present on the different chromosome. No, right? It is not compulsory that the gene should be on the different chromosome. If the gene is on the same chromosome, but it is at the different location, means they are far away. They are far away means obviously on the time recombination is going to be happen. It should be far, not together, very together, means not linked. It should be some distance should be there, far away, or in the same chromosome also. Do you understood, Nili? Recombination will happen on the same chromosome also, but the gene should be in the far away. Okay, finally. So he's taken this character right. Now my question is whether the genes is going to be get the recombination. No, guys. Why? Because the genes are linked, right? They are together only. Together only. When the genes are linked, on the time we will get the a less recombination. Less recombination we are getting means obviously we are going to get a more parental combination. Yes or no? We are going to get a more parental combination. So that's why here, by this experiment, he came to know that, see here, the genes for the eye color and body color were tightly linked. And the only 1.3% of the recombination he got. See here, only 1.3% recombination he got means 99% is the parental type only. Parental, right? 98.7 is parental type only. Only 1.3 percentage is the recombination. Then he did the again second cross. So here what happened? He taken the some other characters. That is a white eye and miniature wing. Here talking the miniature. Okay, miniature wing and the red eye with the large winged male. So characters is different now, right? What he taken now is this is a one gene. Okay, this is a one gene that is having the eye color, eye color, and this is another gene that is going to give the wing shape, wing shape. Okay, and after that he got the result is 37.2 recombinant. Recombinant one he got. See here, Nili, you asked the question right. This these both the genes are located on the same chromosome only. Same chromosome only, but they're not linked. They are unlinked. They are unlinked means there is a far distance between the two genes, right? So in this type, what will happen if the genes are not linked means we are going to get the a more recombination type and the less parental type, less parental type. Okay, but here it is on the same gene only, right? Then also we are getting the 37.2% recombination. Okay, just remember tightly linked genes show the low recombination that we seen in the body color and the eye color, and the loosely linked genes show the high recombination that is the eye color and the wing shape. Okay, do you understood this topic, guys? Can you give me thumbs up if you understood means? The last point which is given, no, that is very, very important. Remember, tightly linked genes show the low recombination. Where the low recombination is there, the more parental combination will be there. Where the uh, where the high recombination will be there low parental type will be there. Okay, fine. So here the same experiment he did. You can see that this is there in the NCERT. That only we explained there separately. So here you can see this is the body color and the eye color. Eye color. So here we got recombinant is only 1.3%. Here 98% parental because these are linked genes. That's why. More parental we got. Less recombinant we got. Then coming to the here what we have taken. Here eye color we are taken with the wing shape. Wing shape we are taken. So in this type, what will happen? We got the recombinant. So because we got the more recombinant compared to this, not with the parental. Okay. Compared to this, we got this one as the more recombinant because it is unlinked. unlinked. Okay. So just remember this. Now go to the next mechanism of the sex determination. How can you do the sex determination means there are the mechanism. So here X and XO type of mechanism. So this type of mechanism we've also called as male heterogametic. Male heterogametic. Here male is going to carry the two different gamete. Two different gamete. Different. Remember, male is going to carry the two different gamete. So here in the X and XO mechanism, the grasshopper is the example. And if they ask about give the example for the male heterogametic means, you can give the X and X, X and XO, X and X and X and XY. These two will give the male heterogametic. Remember this, okay? In the XO and XO mechanism, XX, XX is the female and XO, where only one gamete will be there, that is the X will be there, that is a male. Okay, that is the grasshopper. Then coming to the XX and XY mechanism in the humans and the Drosophila, no need to tell. XX are the females and XY are the male. Then coming to this is the very important you have to remember, that is the male homogametic. Give example for the male homogametic. They will give the option. Option A, humans. Option B, Drosophila. Option B, grasshopper. And option D, birds. Which is the example for the male homogametic? That is the bird. In the bird, the male is going to carry the Z and Z, whereas in the female, it is going to carry the Z and W. Now you tell me, in the bird, who is responsible for the sex determination? In the humans, we know, right? In the humans, who is responsible for the sex determination, guys? In the humans, who is responsible for the sex determination? Male or female? Male, right? But always blame to the girls, right? Male, if for the baby, the blame will go to the female only, right? In the humans. Then what about the bird, guys? In the bird, who is going to be determine the sex of the baby? Who is going to determine the sex of the baby in the bird? Female. Very good, Raju. Very good, Simran. Just remember, the male homogametic example is bird. In the bird, ZZ, ZW. Okay. Okay. Fine. And then coming to the honeybee, you all know that in the honeybee, what will happen? See here, the queen. Queen is there. The queen is diploid. That queen is having the 32 chromosome, 32 pairs of the chromosome, right? If the female is undergone the meiosis, it will show the haploid, right? The egg will be haploid, right? The female lay the eggs means the egg will be haploid. Now, if this egg is undergo the fertilization, if this egg is undergo the fertilization, if it give the diploid, it give the diploid means it will give the female, that is worker. Female worker. Okay. If this haploid will not undergo the fertilization, no fertilization. No fertilization means it will going to be stay like the haploid only. Then it is going to give a drone. Okay. So that's why in the here in the honeybee, the drone is haploid, whereas the worker is the diploid. Remember this. Okay. Okay. Then coming to the mutation, you all know that what is a mutation, right? For mutation is nothing but the sudden heritable change in the DNA. You all know that DNA is made up of the nucleotides, right? Adenosine triphosphate and the guanosine triphosphate, right? These all are the nucleotide where a sudden change will happen to the DNA. That we are going to call as mutation. You know that mutation is going to cause the cancer. Yes or no? It is going to cause the cancer and some of the other genetic disorders also caused by the mutation. So here first we will talk about the point mutation, a change in the single base pair. It is only a point change. For example, so here is the base pairs are there. So U, A, G, 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 here is there. Here the single mutation in the point, this is the point mutation, it is changes the only one base pair. One base pair it is changing means that we are going to call as a point mutation. Example is sickle cell anemia. We will study that where glutamic acid is replaced by valine in sixth position. We'll study about that. It is a point. To the point, the mutation is going to be happen. Okay. And the frame shift mutation, the frame is going to be changed here. Frame. For example, AUA, okay, AUA, AUG, then UUA is there. Okay. What will happen now? If I remove before I am inserting, insertion or deletion will be required for the frame shift. Insertion or deletion. Insertion. Insertion or deletion will lead to the frame shift mutation, guys. For example, I have inserted here one more. Okay. Now, what is the combination it is taking now? AAU. Before it is AUA, right? Now it is AAU. Now it is AAU again. Then GUU, right? So the frame is going to be changed, right? So this is by the insertion and the deletion of the one base pair. Okay. Remember that. Then coming to the mutagens, mutagens is nothing but the agents which are going to cause the mutation. Okay. Here the physical mutagens, if you take, means UV radiation, and you will get the skin cancer mainly by the UV radiation. Okay. And the X-ray, X-ray is mainly going to damage the chromosome. It is mainly going to chromosome aberration. We will call it a chromosome aberration. Okay. Chromosome, it is going to be broken by the X-ray. So that's why the X-ray is not recommended so many times. Okay. Then chemical mutagen, girls and so many we will use the chemical cosmetics and all the thing, right? Due to that also we'll get the cancer. That is also going to cause the mutation. Remember, not all the cosmetics. Some are having some chemicals that is going to cause. Okay. And here the chemical mutagens are mustard gas, phenol, and formalin. Remember the examples. Okay. Then coming to the pedigree analysis, no need to explain this. You know the male, this the round is the female, and sex is unspecified means we will give like this, and affected means we will fill. The mating is means there is a horizontal line between them, right? If it is the mating is between the marriages in the same relatives means the two lines we are going to put, and the here this is about the these are the two parents, and this is the progeny of these two parents, right? The here it is telling the two parents are unaffected and the progeny is affected. It is actually the autosomal recessive. In the autosomal recessive, you will get like this. Okay, that is both the parents are not affected, by the progeny is affected. Okay, then 5 unaffected offspring will be given like this. Okay, so let us study this pedigree analysis. In the pedigree analysis, there will be autosomal dominant trait and the autosomal recessive trait will be there. So here I will tell you the trick, guys. How can you identify the autosomal dominant? In the autosomal dominant, what will happen? The generation is not going to be skipped. No generation skip. No generation skip. Okay. In all the dominant trait, the generation is not going to be skipped. Okay. Whereas in the autosomal dominant, what you can see, both male and female is affected. Both male and female is affected. Just see that. Here you can see female is affected, and here you can see the both male and female is affected, right? Means it is a autosomal. Okay. And here you can see here parent is affected, and here also it is affected. And here you can see this is a parent, here also it is affected, right? This we can tell that it is a dominant where it is not skipping the generation. Okay. Whereas coming to the recessive here, autosomal recessive. First, we will talk about the recessive. The recessive will skip the generation. It will skip generation. Okay. Then coming to the autosomal recessive, both male and female is affected. Both male and female is affected here. Okay. Remember here. See here. Parent is not affected. See here. The unaffected parent is having the affected progeny. See here. Both the parents are heterozygous. Both the parents are heterozygous. They are having the affected children. They are having the affected children. So here also you can see that both the parents are not affected, but they are having the affected children, right? So that is the female and both male is affected here. And the Unaffected in the recessive mainly, in the heterozygous mainly, here you can see recessive. The unaffected parent, unaffected, unaffected parents having affected progeny, having affected progeny. Okay. And here you can see that. So here in the recessive, the skipping generation, the parents are not having the disease, but the progeny is having the disease. Here you can see the parents here 1 and 2. These parents are not having the disease, but here 3 children are there. One of the children is affected. Right? So this is called as the skipping of generation. Do you understood this trick? I think you all know this trick. You understood this trick, guys? I think you know this. It is easy to identify. Can you give me thumbs up if you understood means? Okay, then coming to the genetic disorders, Mendelian disorder and the chromosomal disorder. What do you mean by Mendelian disorder mean? Mendel talked about what, guys? Factors, right? Factors. Mendel talked about the factors, right? What are the factors he talked? Gene, right? Mendelian disorders are related to the mutation. Mutation. If the mutation is caused, that is called as a Mendelian disorder. Okay. Then coming to the chromosomal disorder, this chromosomal disorder is due to the problem of segregation. It is due to the problem of segregation that is in the anaphase. That is in the anaphase. Okay. For example, let us talk about the Mendelian disorder, hemophilia, color blindness, thalassemia, sickle cell anemia, phenylketonuria, cystic fibrosis. These all are the Mendelian disorder. Then coming to the chromosomal disorder, Down syndrome, Turner syndrome, Klinefelter syndrome are the chromosomal disorders. Okay. So, just now I have to tell about the sickle cell anemia. Sickle cell anemia is the autosomal recessive disorder. Recessive disorder. So here what is happening, guys? Here the sickle shape, the concave shape is going to change into the sickle shape. The RBC, right? RBC will be concave shape. It is going to change into the sickle shape. When the RBC is concave shape, it is going to be transport the oxygen efficiently, right? Efficiently transferring the oxygen. But when it is having the sickle shape, it is not going to be transport the oxygen efficiently. Efficiently. Okay, not efficiently. Okay. So what is happening here mainly is you have to remember the genes here. HbA and HbA is the normal, whereas the HbA and the HbS is the carrier sickle cell trait, and the HbS and HbS is the affected one. The affected one will not live. He will die. Okay, fine. So here what is happening? In the sixth position, here you can see on the sixth position, remember this thing, guys. GAG. GAG is replaced to GUG. GAG is replaced to GUG. By that, glutamic acid is changed to valine. Okay. Glutamic acid is changed to valine. So thereby, it is going to cause the shape of this RBC, shape of the RBC to the sickle shape. Sickle shape. Okay. So that is called as a qualitative. Quality of the RBC is going to be changed, right? So that's why it is called as the qualitative. Okay. So, okay, guys, you can go through this PPT. So I tried to cover, but not possible to cover in this 1 hour of the time. Sorry for that. Okay, you can. I will share this PPT with you. This is a very short PPT, and so many MCQs I am added here. See here, so many MCQs are there related to last year 2024 re-examination and the 2024. Kindly go through this question. Okay. And tell me, guys, in the tomorrow's class we are going to do the molecular basis of the inheritance. Do you want me to teach the topic or only MCQs? Because that is also very big chapter, right? If you want me to do only chapter means I will do the only chapter, or if you want me to take a only MCQ means I will do with the only MCQs. Which one you will prefer, guys? If you have any preference means kindly send me a message here. I'll go with that only. Only MCQs or MCQs and both the topic? Okay, okay. Athena is also telling MCQ and Neely also telling MCQ. Helen also MCQ. Both. Okay. Some of the topic I will bring which are very difficult, that topic, and we will solve some MCQs. Yes, 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 Raju. What I will do is I'll bring some high weightage topic and some MCQs also, which are very asked in the previous, that is 2024 exam. Okay. Okay, Simran. Some topic, highly important topic I will explain, and I will explain this thing. Okay. I think in the today's class I have explained all the important topics. You have to go through with only the disorders or the disease, right? You can remember that. Remember, guys, 4 to 5 questions you are getting. Don't neglect this chapter. Okay. Fine. Okay, guys, I will go for the 1 minute. Yeah, I will go for the this thing. You can give me your feedback. Okay. 1 minute. Yeah, if you have any still questions means you can ask me. Okay. 1 minute. Yeah, I'm launching the feedback thing for you. You can go through that. In the tomorrow's class, we'll go with the MCQs and again that. Okay. The chapter is molecular basis of inheritance by in the tomorrow's class. Shreya. Okay, guys. Thank you so much. Thank you, Simran. Thank you, Nabia.